Well documented as a botanical medicine for over 5,000 years, the discovery of endogenous cannabinoid receptors and evidence-based reports indicating the efficacy of medical cannabis for treating pain have renewed consideration of its therapeutic use. The past quarter-century's advances in healthcare science has included the discovery of a previously unknown human endocannabinoid system dedicated to maintaining critical balances between our mind and body. The following recording derives from a 6-part series of sequential lectures concerning endocannabinoid receptors and medical cannabis, as narrated by physician expert and author Dr. Jeffrey Block. This publication emphasizes how an appreciation of the biologic context to show how and why endogenous cannabinoid receptors function allows healthcare providers to better interpret the limited evidence-based data. In order to properly assess the abundance of anecdotal reports concerning cannabis, the safe and effective treatment of pain will require careful clinical monitoring of patient outcomes to establish and validate the evolving evidence-based data. Dr. Block is an American board-certified anesthesiologist and respected thought leader with a clinical scientist's understanding of the healing potential found with certain botanical medicines. Fellowship trained in both pain management and addiction medicine, his pragmatic rationale considers a citizen patient's compassionate access to the cannabis plant's bioactive chemicals when used for the treatment of pain, as well as acknowledging cannabis's obvious public health concerns ranging from contaminants to dependence. The author's insight shares contemporary perspectives by first acknowledging the bioethical obligation healers have to not simply feel compassion for their patients, but to strive to effectively treat pain. This is emphasized while at the same time acknowledging those concerns to public health surrounding addiction and accidental overdose deaths. Dr. Block's writing reveals how and why a human being's natural instinct nurtures their nature through their body's own endocannabinoids, and why a contemporary physician healer's understanding of the traditional ethnobotanical role certain plant-based medicines have played in treating the intolerable symptoms of disease appears to have also tangible value in modern healthcare. As read from Endocannabinoid Receptors and Medical Cannabis, here is the author Dr. Jeffrey Block's narration of Section 3, The Cannabis Plant's Chemical Complexity.
Delta-9 Tetrahydrocannabinol, or THC, and Cannabidiol, CBD. Delta-9 Tetrahydrocannabinol, or THC, has been the focus of considerable inquiries that have produced indications of its efficacy for managing nausea, appetite loss, and of particular interest, pain. Like anandamide, THC is a partial agonist of cannabinoid receptors. When compared to THC alone, the collective effects of several cannabinoids, terpenes, and other chemicals in the whole cannabis plant has been consistently shown to produce superior perceived effects when compared with cannabinoid isolates such as THC or synthetic cannabinoids, both in terms of maximizing efficacy and minimizing side effects. This is often referred to as an entourage effect, but that term can be a bit misleading in that it implies that THC is the star or leader, and other chemicals contributing are hangers-on. A more accurate way to refer to this chemical synergy might be as an ensemble effect, as the whole plant's constituents work in concert, often without THC itself coordinating the full balanced orchestration. This presents significant clinical difficulties in that the total components of this ensemble have yet to be uniformly researched, and the FDA requires drug makers to follow good manufacturing practices to deliver a reliably consistent, known dose of bioactive chemicals. Whole plant botanical cannabis products that contain many chemicals are exponentially more challenging to study than medicines that contain known amounts of only 1 or 2 cannabinoids. Cannabidiol, or CBD, has received considerable recent attention as a potential therapeutic agent for a host of conditions. Since CBD is not intoxicating, it may offer a more favorable side effect profile than medicines containing THC. A recent critical review of CBD by the World Health Organization's Expert Committee on Drug Dependence concluded that CBD poses no risk of abuse or dependence and recommended that pure CBD extracts not be subject to scheduling. The World Health Organization's expert determination on CBD mirrors that of the FDA experts who evaluated the CBD medication Epidiolex and concluded that its safety profile and lack of abuse indicate that it should not be scheduled as a strictly controlled substance. CBD is a negative allosteric modulator of CB1 receptors, meaning that it modifies some but not all of a cannabinoid receptor's normal functions. For instance, even though CBD does not activate CB1 receptors directly, it can slow the rapid degradation of the endocannabinoid anandamide to prolong and enhance its natural effects on CB1 receptors. As an allosteric modulator, CBD is capable of attenuating some of the dysphoric side effects from delta-9 tetrahydrocannabinol, such as paranoia. Another allosteric modulator, the endogenous hormone pregnenolone, also produces an allosteric inhibitory action on CB1 receptors that reduces the psychotropic effects unique to THC. Pregnenolone synthesis increases in response to THC administration, and exogenous administration has been proposed as a method of mitigating THC's central side effects. Similarly, CBD modulates the effects of the partial agonist THC on CB1 receptors, inhibiting some of the intoxicating effects of THC by adjusting anandamide levels and modifying adenosine uptake. CBD also appears to inhibit the digestive metabolism of THC edibles that through liver metabolism produce the considerably longer-lasting and potentially more potent psychotropic metabolite 11-hydroxy-THC. At CB2 receptors, CBD can function as a partial agonist, not fully activating the receptors but attenuating them, much as THC does at CB1 receptors. Phytocannabinoids such as CBD have many targets other than CB1 and CB2 receptors, and CBD may be considered as the epitome of polypharmacy. It can interface with a host of other enzymes, transporters, ion channels, ligand-gated ion channels, GPCRs, and nuclear hormone receptors. CBD can stimulate adenosine receptors and provide indirect pain relief by modulating other receptors indirectly. Perhaps most significant among these for CBD and pain may be the transient receptor potential or TRP channels, particularly those designated as transient receptor potential vanilloid 1 or TRPV1 channels, which appear to contribute to the control of inflammation, pain, and temperature sensation. Both CBD and anandamide can stimulate TRPV1 channel activity. The extent to which CBD appears to activate and over time may desensitize TRPV1 through receptor downregulation helps to explain CBD's analgesic and anti-inflammatory effects. Other minor phytocannabinoids, but not THC, also appear active on TRPV1. CBD seems to function in diverse ways, stimulating receptors, enzymes, and other proteins as an agonist, antagonist, and allosteric modulator. CBD also acts indirectly by inhibiting the degradation of endocannabinoid system components such as anandamide and other neurotransmitters. When administered alone, CBD may be acutely ineffective at low doses of less than 200 milligrams per day, while CBD administered in larger doses, for example between 200 and 800 milligrams per day, can produce many discernible therapeutic effects. Those effects can be observed at significantly lower doses when administered in combination with THC. Lower doses of CBD and THC in a 1-to-1 ratio can produce comparable effects to high doses of CBD in isolation, as noted in relation to seizure control. CBD modulates effects of THC through direct and indirect mechanisms. In addition to augmenting anandamide levels and modifying adenosine uptake, and without causing sedation, CBD can counteract paranoia and anxieties that result from THC exposure, particularly in cannabis-naive users, by activating complementary receptor functions that have anxiolytic effects. CBD also potentiates the amino acid tryptophan, found in turkey and other foods that are known to be associated with elevated mood and relaxation. All of these can be positive effects in patients with pain who often suffer associated anxiety and dysphoria. Rodent studies, anecdotal reports, and data collected by cannabis product manufacturers provide evidence that CBD may be effective in both topical and systemic treatments for certain localized pain, particularly arthritis. Research has shown that the joints of rheumatoid arthritis sufferers have a high concentration of responding CB2 receptors, consistent with their on-site, on-demand protective role in fighting inflammation and injury. Some arthritis sufferers report that their disease progression slows or ceases even with topical cannabinoid medicine use. The United States Food and Drug Administration's approval of CBD as a medicine. Most prominent among the potential applications of CBD has been its use for seizure control. Plant extracts producing CBD have been shown in clinical trials and numerous anecdotal reports to mitigate seizures associated with several disorders, particularly among refractory patients. The U.S. Food and Drug Administration approved Epidiolex in 2018 as a standardized dose control CBD medication manufactured from cannabis plants. It was approved only for Dravet syndrome and Lennox-Gastaut syndrome, two rare pediatric seizure disorders following clinical trials that were facilitated by an orphan drug designation that provides incentives for the development of drugs to treat rare diseases affecting only small populations. Subsequent to the approval of Epidiolex, the DEA issued a scheduling order placing only FDA-approved drugs that contain CBD derived from cannabis and no more than 0.3% tetrahydrocannabinol in Schedule 5, the least restrictive controlled substance category. Currently, that designation exclusively applies to Epidiolex as the only approved CBD drug and does not impact Schedule 1 status of any other CBD product. It is nonetheless a significant milestone in cannabinoid medicine development and opens the door to similar plant-derived cannabinoid isolate drugs that may achieve FDA approval for these or other conditions. The approval and scheduling of Epidiolex has contributed to confusion over the legality of other CBD products. Many companies are aggressively marketing CBD products as legal to ship to all 50 states, which they are not as the time of this publication. Some claim that because the Controlled Substances Act of 1970 identifies THC by name as a Schedule 1 substance but makes no mention of CBD, CBD is not scheduled. Others claim that because the Controlled Substances Act distinguishes between Schedule 1 marijuana and hemp, a varietal of the cannabis plant containing less than 0.3% THC, any CBD derived from hemp is legal. In 2016, the DEA rejected these arguments and published guidance, noting that phytocannabinoid extracts will remain covered by the Controlled Substances Act and that any plant materials from which more than negligible amounts of cannabinoids can be extracted is by definition marijuana and not hemp. In fact, the testing of products marketed as hemp-derived CBD extracts has revealed that most of them contain little or no CBD. The Farm Bill of 2018 may facilitate access to CBD derived from hemp as it contains language to exclude hemp-derived CBD from the Controlled Substances Act, but the bill does not affect the scheduling or FDA rules. Additional bioactive chemical components in cannabis. The total cannabinoid content of a product offers only a crude index of therapeutic effect. since much of the end-user experience is dictated by other chemicals acting in concert, including the 100-plus cannabinoids in plant material and abundant terpenes. While the ratio of cannabinoids, particularly between THC and CBD, may help predict many effects, terpenes figure more prominently than was realized until very recently. Of interest in relation to the clinical trials conducted in the development of the standardized dose-controlled plant extract medicine nabiximols is that the approximate 1-to-1 ratio between THC and CBD was demonstrated to be most effective for analgesia. That balanced ratio is characteristic of some historical landrace strains of cannabis cultivated over thousands of years, but as the illicit recreational market developed, that ratio would tip dramatically to the intoxicating THC as the intoxicant modulating CBD and other terpenes were recessively bred out of commercial cannabis. The development or reclamation of CBD-rich hybrids of cannabis has been a renewed interest for contemporary medical cannabis cultivators. Medical cannabis dispensaries and product manufacturers still mistakenly label their products based on a perceived distinction between different species types of cannabis plants, with indica or sativa respectively identified as being either sedating or energizing. While there is an historical basis to the evolutionary development of plant phenotypes, indica cannabis varietals developed in northern latitudes, while sativa types came from equatorial regions, recent genomic analyses of cannabis plants reveal that contemporary plants should be essentially regarded as hybrids of the two, with little chemical distinction in terms of their cannabinoid profiles. Where pronounced differences appear is in the plant's terpenes. Terpenes are the bioactive chemicals that give all plants their smells. These and other chemicals can influence receptor activity, including analgesia. An example is the reported sedative effects of some CBD-dominant cannabis hybrids. While CBD has demonstrated anxiolytic effects that may contribute to relaxation, hypnotic sedation is typically associated with the terpene known as myrcene, which is often abundant in CBD-dominant varietals having both sedative and analgesic properties. Myrcene is the likely source of what is a colloquial term called couch lock, a perceived inability to engage in physical movement or activities that appears to result from myrcene's hypnotic effect. Other terpenes and cannabinoid profiles can produce a range of distinguishable effects. This explains why patients often tend to prefer particular varieties, types of products, or methods of delivery, and why availability of a wide variety of products can be helpful.
This recording has been a healthcare educational initiative read by physician-author Dr. Jeffrey Block. Dr. Block is the founder of Nurturing Nature Group Consultants, an independent and scrupulously neutral interdisciplinary consultancy group advancing endocannabinoid science through translational research within the practice of integrative medicine. To learn more, please visit nurturingnature.com.